I know, it would have been interesting. But now we're going to start with the biosynthesis of amino acids, a little bit with nucleotides, not a whole lot, and some of the other biomolecules. And to begin that, we actually start with nitrogen, because we've already discussed how to get rid of nitrogen, but we actually need to know where it comes from. From where does it originate? Because behind only carbon, hydrogen, and oxygen, it's number 4 per mass in all of the living systems. So we actually do need a lot of nitrogen, but we also know that it's a little difficult to get rid of it. So where does our nitrogen come from?
Amino groups.
Right. So, but I mean, where from? I mean, we don't make nitrogen. Right. You're going to have to consume it. I mean, we don't have a way to utilize it from the air, even though 80% or more of the air, is into. We don't have that ability to fix the nitrogen. All right, certain amino acids and or portions thereof, for example, well, we'll go into that later. They are incorporated into nucleotides, and so much of the chemistry behind amino acids and nucleotides are interrelated because there's a transfer of nitrogens. There's the one carbon transfer groups, which we've alluded to in the past. For example, methionine becoming homocysteine because it can donate that terminal methyl group that's hanging off that sulfur. Okay. And so, by the way, if you ever forget like what methionine looks like, just think it has that extra methyl group from homocysteine. So you just need to know what cysteine looks like. You add an extra CH2 linking group, that's homocysteine, and then you add the methyl and that's methionine. And we'll talk about homocysteine in greater detail later on because it's also important. And then there are lots and lots of genetic diseases related to these various pathways with respect to amino acids and nucleotides. So this is the nitrogen cycle, and I don't know if in biology do they cover the nitrogen cycle in any detail? I mean, I know it's been a long time ago for you guys, but actually, whenever I took biology, they hadn't even discovered Animox bacteria. So the nitrogen cycle that I learned about was different than this one, but we're going to talk about how it is. It's just like here, just because just like with carbons, nitrogen can exist in different oxidation states. And so another easy way to look at it is that the more bonds the nitrogen is making to an oxygen, the higher the oxidation state. It's being oxidized. If you decrease the number of oxygen-nitrogen bonds, you're reducing it. Or if you're increasing the number of nitrogen to hydrogen bonds, you're reducing it. So that's why ammonia, or ammonium, since that's the way it would exist, in vivo, is the most reduced. Whereas nitrate, NO3 minus, is the most oxidized. Okay? So we're going to break it down to the different parts in a little bit of detail here. I don't go into a lot of detail, but there's some. Let's see. Okay, so first up, as I mentioned before, 80% of the air is N2. It is not oxygen. By this point in time, you probably already know that. Maybe not in biology 1, but the vast majority of your air is N2. It's inert, and so you don't have to worry about it. But our body, we can exhale it, inhale it, exhale it. Unfortunately, we don't have a way to fix it, to put it into our, amino acids and nucleic acids, so therefore we have to have another way around that. So plants, because of the nitrogen-fixing bacteria and archaebacteria can incorporate into ammonia, plants then take the ammonium and they incorporate it into amino acids. And so that's why either we have to eat the plants and get the amino acids, or we have to eat the things that eat the plants to get the amino acids. Okay. The denitrifying bacteria, certain archaea and fungi, they actually utilize nitrate instead of oxygen to make their ATP, and we're going to show that in just a little bit. Okay, so that's really, in a moment when we talk about the Animox bacteria, it's really cool because hopefully you'll be able to see how it correlates to our OXPHOS pathway, but the fact is that they use nitrates instead of oxygen. And so, we are blessed in that sense. It's a very symbiotic relationship between the little critters and us. I'm sure that microbiologists just love me calling it critters. And then relatively recently, I don't remember the exact years, like 90s, early 2000s or so, they discovered that there are a type of bacteria, or there is a type of bacteria called the Anammox bacteria, and they think that they convert anaerobically more than half of the biosphere of the N2 from ammonia. And so they actually help make the nitrogen that's in the atmosphere that then get used by other bacteria to make the ammonia and to help keep the cycle up. And they can also utilize nitrite, which is good considering the fact that nitrite and nitrate are both oxidizers. Okay, so we don't want to have too much nitrite or nitrate around. So those are the anammox. They use something called an anamoxosome. You know how biologists and cell biologists love that zome ending. Okay, so the anamoxosome is a special compartment. Okay, and what it is it's really cool because they do take from their cytosol, they incorporate ammonia, and then they utilize this compartment called, or this enzyme called hydrazine hydrolase, which, as we know from hydrolases, you're adding in, well, you utilize water, but to add in the hydrogens, and they make as their inner compartment, so it would be similar to the matrix, and I don't know if they call it the matrix in the anamoxosome, but what would be similar to the mitochondrial matrix there is they make hydrazine, which actually is rocket fuel. Okay, which is kind of ironic, I think, because you find these a lot of times in sewage, so it's an explosive. Okay, and that's an intermediate where then we have this hydrazine oxidizing enzyme complex that helps, that takes the place of the electron transport chain, and ultimately it's nitrite instead of oxygen that gets utilized and transformed into water and or the ammonium hydroxide. Okay. Hydrogens get pumped across, and then once again they have an ATP synthase. And that's how they make their ATP. Okay, and so but they use this hydrazine and they fix the nitrogen to where one of the byproducts is N2, and therefore it gets excreted out into the atmosphere, which then other organisms can utilize that to make nitrogenous compounds that can be utilized by plants and other bacteria.
Would you expect us to draw a cartoon?
No. Other than you just know that it's in the anamoxosome, and that takes the place, essentially, of the electron transport, OXPHOS pathway. ETC OXPHOS pathway, because it is an oxidative phosphorylation pathway, it's just not utilizing O2, it utilizes nitrites and produces N2.
So N2 is the final.
N2 would be the final recipient, or final product there, similar to the way that we have O2 to water. Or I should say that's one of the final products. They have multiple products. Okay. It is very exergonic, as you can imagine, but the N2 is stable. So because of that, it has a very high energy of activation. So then what happens is certain bacteria and archaea will utilize ATP, since N2 is so stable, and to break it down. And they have a whole complex that I do want you to be able to discuss called the nitrogenase complex. So nitrogenase complex is what's going to ultimately be breaking it down. Okay. And it's a series of oxidation reduction reactions. I've got pictures. They're cartoony kind of pictures coming up.
It's breaking down.
Oh, I'm going to show you. It breaks down into, so I mean, because it's going to break down the N2 to ammonium, well, essentially ammonia, but ammonium, which can be utilized by other organizations. And it does so really at the expense of pyruvate and acetyl-CoA to acetyl-CoA, similar to what we've seen in the past. So if we look there, we can see where the pyruvate gets broken down to acetyl-CoA and carbon dioxide. There is some variation, but at least one species is pyruvate, I should say, the one that's most widely studied. Okay. And you're going to have some type of ferredoxin or flavodoxin, which we've seen. These are like the FMN type proteins. And so these cartoons just go from, the oxidized to reduced state and reduced to oxidized because they just pass the electrons off from one, I don't want to say species, but from one electron donor to the next electron acceptor. Okay. All right. Oh, where are we going now? There we go. Okay. So if we start with pyruvate going to acetyl-CoA, and it does 8 electrons at a time, but I'm not worried about the numbers so much. You have the oxidized flavo protein becoming reduced because it's gaining those electrons, but then it turns around and it gives up the electrons to the next item in the next enzyme in the list, okay, which is called dinitrogenase reductase. Okay. And it does require ATP here. So, because it's going to take a lot of energy to break down nitrogen. And we're going to go in a little bit more detail about the dinitrogenase reductase and dinitrogenase in just a moment, because it utilizes something that we haven't seen before, I don't believe, at all. Okay, so a new metal is going to come along that's really important. Okay. So now the dinitrogenase reductase, it's been reduced. It has to go through phosphorylation, and it's going to pass off the electrons from the dinitrogenase reductase. What do you suppose it's going to be reducing? What does this reduce? Dinitrogenase reductase. Just based off of its name, what do you suppose it's reducing? No, not even in 2. It's something even more simple based off of its name. Dinitrogenase reductase. So it is reducing dinitrogenase. Dinitrogenase is the enzyme that does the atmospheric nitrogen. Okay. So dinitrogenase goes from the oxidized state to the reduced state, and then it can go on and reduce the N to ammonium. But what's really cool about dinitrogenase, it is a tetramer, but it contains and it utilizes molybdenum. We haven't seen, I don't think we've even seen molybdenum before. We haven't really talked about it. It is a trace element that's required by certain species. Even us, we have some molybdate requirements ourselves. Now, there are some species that actually use vanadium, which we don't even talk about that very often. And by the way, vanadium does look an awful lot like vanadate, looks an awful lot like phosphate. So it's one of the reasons why sometimes it's an inhibitor of various things that requires phosphates. Okay. But most of them utilize iron and molybdenum. And I'll show you, if you look at the numbers for the molybdenum-containing enzymes, there's 2 molybdenum, but there's 32 irons, 30 sulfurs, and it requires homocitrate, which is a cofactor that we haven't talked a lot about, actually at all. We talked about citrate, but not homocitrate. So I'm going to show you a picture in just a moment of what this all looks like. So I hope you can tell the colors. They're the dark ones and the light ones to me. Now I can tell the blue. So I know the blue ones, the blue and the green ones are the reductase. So the nitrogenase and the dinitrogenase and the dinitrogenase reductase actually form this huge complex. And so the blue and the green are the reductase, which should be on the outside, so to speak. So that's why it would make sense that they get reduced first, and then they go in and they reduce. They don't go in, but they pass the electrons off through, and I'll show what these look like in just a moment, the molybdenums and the irons in order to go ahead and reduce the dinitrogenates, which is on the interior of the complex. Okay? ADP is in red, iron's in orange, sulfur yellow, molybdenum in black. And so I'm going to peel away the proteins in just a moment, so that way you can see the way that the metals line up. So there we go. That's the iron molybdenum cofactor. So since we've peeled this away, now we can see where we can actually have the electrons get passed through, that, whoops, it's a little slow today, but get passed through along that chain, essentially. To the interior of the complex, to where ultimately it's made into NH4 plus. Okay. So this is just a look to see, where we have the molybdenum and the iron, how that they get interrelated, and it goes along. So they are very close. It's very similar to the iron-sulfur proteins that we had talked about in the past, except this one just it has a molybdenum in it. And molybdenum is a transition metal. We don't have a periodic table in here, but if you remember vaguely where it's at, it is located in the transition metals, and so therefore it does have multiple oxidation states. Okay. And of course, the higher the positive number, the more oxidized it is. Okay, so now we're going to look into how the ammonia produced by these bacteria and archaea get incorporated into biomolecules, and what possible amino acids that get used for this. What kind of amino acids would make sense if you're going to use an ammonia. So we're going to be talking about things that have nitrogens in them. Okay? It's glutamate and glutamine, which is very similar to the way that we get rid of the nitrogens. They are the ports of entry for the nitrogens. Okay? Most organisms contain an enzyme that will take the ammonium and make it, well, I shouldn't say make it and add it to something in order to synthesize glutamine. Okay? So what classes of enzymes do you suppose can do this? Like, what would be some of the ones that would make sense? Like synthases, synthetases, stuff like that. Okay, so this is a two-step reaction, and it takes glutamate and ultimately converts it into glutamine. Okay? Because it does require energy, because the glutamate, if you think about it, there's already resonance due to the fact that it's a carboxylate, and so it's actually lower in energy than a glutamine, which has less resonance, since it's an amine in that terminal group, and so we have to use up an ATP. And this is a gamma. That means it's adding it to the end, the gamma position. So glutamate becomes gamma glutamyl phosphate, and of course ADP would leave. And then, since that phosphate's a great leaving group, the amine can go in and attack it, and you release the inorganic phosphate, and you now have glutamine. So as you can see, it's a large complex. And as we mentioned before, it would be the synthetase here, since it utilizes the ATP. Probably would have helped if I would have told you to utilize ATP first, but my bad.
Where does the enzyme come?
Oh, the enzyme is what does both of these steps. So it would bind the glutamate first, and the ATP. The ATP, pardon?
So it all occurs at the
Right. Mm-hmm. It all occurs within the enzyme. It's a ping pong mechanism where first the ATP and the glutamate bind. The phosphate gets transferred to the ADP leaves, and as the ADP leaves, the ammonium comes in, kicks off the inorganic phosphate, and then glutamine leaves. So there is that enzyme intermediate that is bound. Okay. Now, plants and bacteria can synthesize glutamate from glutamine using an enzyme called glutamate synthase, but we cannot. Okay, and so this one, if you notice, since it's a synthase, it does not require ATP. It utilizes our good friend alpha-ketoglutarate. And so we have an alpha-ketoglutarate and a glutamine, and you get two regular glutamates at the expense of an NADPH. Okay. And so what's really cool about this is if you take the previous reaction from the glutamine synthetase and you take the glutamate synthase, you can actually yield a glutamate from an alpha-ketoglutarate. So you can completely forego the aminotransferase that plants and bacteria can. We can't. We always have to have that. If you remember, the alpha-ketoglutarate becomes glutamate and using the aminotransferase, and it's that way with all the aminotransferases. Well, plants can actually get around that by making a glutamine intermediate. It's just a second. There we go. Okay, so glutamine synthetase is the primary regulatory point for this. It's regulated in all organisms, and it's regulated by many things. So I like to think of this as like the kind of light switch that's the dial versus just either on and off. Because a lot of times when you talk about like phosphorylation or something, it's either activated or it's inactivated. So it's an on and off type of light switch. This one, since it has so many, it has 8 different allosteric cofactors that can either inhibit or activate it, then the fact that it's additive, then you can make it less off than at other times. So you can either completely shut it down or just down-regulate it, to 50% or 20% or what have you. Okay. But it's alanine, glycine, and then 6 of the other end products that require glutamine. So here we go. Like I said, there's a lot. You can see there. It's a huge complex.
So do you want us to know all the inhibitors and why, or just?
Well, some of these should make sense. Okay. I hate to say that I just want you to memorize them, but I would at least expect you to know the rationale behind them. Okay. Glycine and alanine are not made from glutamine, but a lot of the other ones, all these other amino acids are made from glutamine. Okay, and so then it should make sense that if we have, for example, too much, come on now, I don't know why it keeps freezing. If we have too much histidine, which comes synthesized from glutamine, that means that we have plenty of it. We don't, that you don't need to make any more glutamine. So it's going to go up and it's going to down-regulate. All these are inhibitors. Okay. Same thing goes with tryptophan. AMP is due to the hydrolysis of ATP. So if you have a lot of AMP, that means you don't have a lot of ATP. So you're going to want to be able to resort it and turn this off or downregulate this so that way you can start to synthesize ATP. Otherwise it would just completely deplete it. Why does carbamoyl phosphate kind of make sense? Where have we seen carbamoyl phosphate? It's in one of the pathways that we've seen so far.
Isn't that the B12?
It's not for B12. What metabolic pathway has carbamoyl phosphate? Urea cycle. Remember, that's sort of like the step 0 or step -1 or whatever you want to call it in the preparation, because then we have the transferase that comes in and adds it on into ornithine. And so if you have a lot of that, then that means you do not need to be making or putting in more nitrogen, you're trying to get rid of it. Okay, so now glucosamine 6-phosphate is something that's new. It's a sugar derivative, but otherwise, all these should make sense. And the CTP is because it's made from nucleotide. It's one of the nucleotides is made from glutamine. So if you have a lot of CTP around, that means you don't need to be making more glutamine. That is made. That prerequisite has been covered.
What is
CTP, cytosine triphosphate. You know, it's used to make RNA. If you make it deoxy, it'd be used to make DNA.
So glutamate is made into glutamine if you need one of the 6 things.
Right.
And it
If you
only
need
does
one, or
at time.
typically, usually you may, for example, the amino acids, you may need more than one of those amino acids, right? So therefore, we're going to convert glutamate, which we have a lot of glutamate around. Then we're going to take it away from the aminotransferases, use it to make the glutamine, and the glutamine is the next step to make these other amino acids and nucleotides.
But like, let's say you have a lot of tryptophan, but you don't have a lot of histidine.
Then,
So-
right. So then then the enzymes would be able to kick in to make more histidine.
Okay, but the tryptophan wouldn't inhibit it, even though.
The tryptophan could inhibit it, but it wouldn't inhibit it completely. That's where you just say, instead of running it at 100%, we only need to run it at 87% or whatever. I'm making that number up, but you can just downregulate it a little bit than having it go gung-ho. Whereas if you need both tryptophan and histidine, then it's not going to be as inhibited. Maybe it's running then at 92% or something.
Why is the glycine and alanine inhibited?
Those, usually if you have excess of them, then you're going to have excess of other amino acids too. Plus you can also think if you have a lot of alanine, then you are already at high energy. And we're going to see glycine is actually a precursor for other. Well, we already know that alanine can be used to make lots of things. Well, glycine can too. In fact, it's amazing what, I always find it really cool what glycine is used to make because it doesn't seem to be the obvious choice, at least in my personal opinion. Of course, I'm not God, even if I sometimes act like I am. Just kidding. All right. So not only those are allosteric, it can also be covalently regulated. Okay. So first off, it takes me a while every year to look at this and try to figure out what all's going on because this one is not the easiest figure. So I would suggest that you mull it over after when you read this part of the book. First of all, the enzyme itself contains an amino acid that can be phosphorylated. So if you look up there, what amino acid could this be? This is and technically it's not just phosphorylated, it's AMPylated, adenosine monophosphorylated. Yeah, AMPylated, that's what I was, ampylated. It's kind of hard for me to say. And AMP gets added to that amino acid. What amino acid is that? There's one that's even more sense than tyrosine, right? Because there's an alcohol that can go in and smack the ATP at that first phosphate and knock off the inorganic pyrophosphate. So yeah, it's a tyrosine. Phenylalanine doesn't have a nucleophile on it, so it'd be more difficult. Oh, whoops! I was really kind of hoping that it would go through. It does increase the sensitivity to these allosteric effectors, and the complex itself has a regulatory protein that's called P2. Now, I don't know where the best place to start what this is. So, ultimately, our goal is to look, here, there's it's slowly moving. It's like stunted right now. So we're going to look at the inactive versus the active form, and this is only for the covalent regulation here. Okay. So you have those 8 things that combine to help downregulate it. You can also downregulate it, make it inactive by AMPylating it. I really have problems saying it's called adenylation, you add an AMP. Okay, so if the AMP is on it, that tyrosine is inactive. Like that nucleophile is gone until you can regenerate it as a free alcohol. So then removing is called de-adenylation.
You said you're adding an AMP to it.
Yeah, that's what this is showing you. This is AMP up here. It's been added on to that alcohol. Okay, so let's. Okay, this is where it gets really hard for me to talk about. AT is adenylyl transferase. Okay, instead of it being an amino transferase, this is the cartoon is showing it as an AMP transferase. Okay. It is activated by P2 whenever P2. Let me see if I can do it this way. I wish it would just let me make it bigger. Will it not make it bigger on yours? No, I guess it just makes it bigger on mine. Never mind. Okay. So P2, if the P2 hasn't been umped, because that's a uridylation. I'm just done doing years and years of speech therapy as a child. So growing up when I was little, I had a speech impediment where I sounded like I was from Boston or New Jersey or something like that, which is fine if you live there, but not good if you live in the Ozarks. Okay, so.
Jerseys that aren't good in America.
Yeah. So, yeah, I haven't been impressed anytime I've been to New Jersey. But yeah, like I had a bird named Archie, a cockatiel, and so I'd say, I got a boy named Archie. You know, we go caw the caw, that kind of thing. And so that was not very good. So saying stuff like uridylation is really hard for me. Okay, so I'm going to call it umping. So what happens is the P2 can be umped and have that UMP added to it by this enzyme, UT. What do you suppose the enzyme UT stands for?
University of Texas.
Ha ha ha. God, no. So, other than Ms. Burke, what does UT stand for? Exactly. Mm-hmm. And so, it too is regulated, but it takes a UTP. Oh, whoops, I didn't mean to cut it off. It takes the UTP and it adds the UMP to the P2. And that P2 still binds to the ATP and it can actually then de-adenylate. Okay? So that's why P2 actually has two roles under two different conditions. And you have this UT enzyme that can either be activated or deactivated as needed. Okay? All right. So whenever the AMP is added, it's to the glutamine synthetase, it's turned off. Whenever it's removed, it's turned on. So the way that this happens, one way is if glutamine is made, so we have a lot of glutamine, it actually is going to go back and do a feedback inhibition. Because we don't need any more glutamine being made. Whereas if you have a lot of alpha ketoglutarate, it has the exact opposite effect. Okay, because then we don't necessarily have a lot of glutamine.
So when do you have the covalent regulation versus the allosteric?
You have them going at the same time. It's just the allosteric, it would require all 8 conditions to effectively completely turn it off. This is, I guess, another quicker way to completely turn it off if you want to think of it that way. So this one is more like the light switch versus... Okay, I still have almost 5 minutes. So this is more like the regular light switch, because it's either covalently modified or it's not for that tyrosine, versus just down-regulating it fractionally. Okay. So at this time, I think I'll end here because this is when we're going to start talking about using SAM